So what is this mega bombshell news that Pfizer mRNA vaccine could be causing this phenomenon called frame shifting, which basically would mean that it could lead to production of gibberish proteins. So that just came out recently in an extremely prestigious scientific journal, so you can't really discredit it easily. So we're going to talk about this, and we're going to be talking about potential consequences of that, if any. And because as it happens, I just happened to be involved in another scientific review, which was just on that very topic. So that means I just happened to be able to have a discussion on this very information, which we suspected might be the case, and we just got a confirmation of this. So we're going to address all of that in today's video. It's going to be a little bit lengthy video because of that, because there's so much to cover. My name is Dr. Mikhail Rashik of Genomics, and let's get going with this. And I can tell you, there is definitely bad news in that this frame shifting has been observed. So there's no sugarcoating this. We don't know yet what the biological consequences of that might be, but it does seem to lead to production of unwanted proteins, basically new constructs that were not meant to be produced. So that is definitely real. There's no denying it. So we'll see what that means. And another thing is, I can tell you what I wanted to do is we're going to get into detail of this paper, because one thing that I really enjoyed is that this publication is how it was discovered. It's quite ingenious. So we're going to be talking about this. But my favorite part of this paper is that the authors also provided a solution. So it's an easy fix to this problem, which is fantastic. This is probably one of the most valuable papers we have yet received this year that provides easy solution for the vaccine industry on how to fix this problem. So of course the question is, why did this problem even exist in the first place? And it's the fact that no one has ever yet looked for that possibility, for the possibility of this frame shifting taking place with such novel technologies that we applied in the production of these mRNA vaccines. So to the author's knowledge, this was the first paper of its kind that demonstrated this. Hence, this is what happened. But the question is, who is to blame? Well, that's a tough question to answer. So we'll talk about that as well, if anyone, for that matter. All right. Okay. So first, let's talk about what is frame shifting. So really what we're talking about is, remember, when you produce mRNA based on genetic information such as the DNA we have inside our nuclei, the DNA is just a repository of information, if you will. And you use that information to select what kind of genetic blueprints you will produce. Those are called mRNAs. This is what we did with the vaccines. We simply used mRNA that is not found inside our cells, but we basically seduced our cells to produce specific protein of interest. In this case, that was the spike protein. So mRNA is the template, and then that template has to be recognized by specific machinery that then uses this template to produce what we really want, which is the protein, such as the spike protein. That was the purpose of the mRNA vaccines. Now, when that machinery, which is called ribosome, by the way, uses the genetic template, the mRNA template, the way it knows how to produce a protein is by looking at the genetic code. And it looks at that genetic code 3 nucleotides at a time. So nucleotides are chemical bases. There's 4 of them. That's basically how you code the information. And you look at the genetic information 3 nucleotides at a time. That tells you which amino acid you're supposed to be incorporating during the production of the protein. So 3 genetic codes means 1 amino acid. You move to the next 3, that's the next amino acid, and so on. As a consequence, this process is called reading in frame. You're reading the genetic code, the mRNA, in frame. If that machinery, that ribosome, accidentally moves by 1 or 2 of those nucleotides instead of 3 ahead, then suddenly it will be reading out of frame. And as a consequence, now you're producing something completely different because it's not the code you're supposed to be decoding. Now you're decoding aberrant information, aberrant code, and that is what is referred to as frame shifting. And this is how you can now, as a consequence, produce gibberish protein. So fragment of it might be okay, might be real as intended. If the machinery moves out of place, then after that event taking place where it moves out of place, it literally produces gibberish, hence the frame shifting. Now, frame shifting, that can be taken advantage of in natural processes. So viruses, because viruses are so tiny, they need to bring as much of their information as possible in their genetic code, so they can actually employ this strategy where in a very small amount of genetic information, you can produce more than 1 protein. So one way you can read the protein in 1 frame, and then if you have the machinery move by 1 extra place and read the genetic code, it could produce completely different protein. Quite an ingenious way for nature to be able to encode information in such a way. And apparently SARS-CoV-2 virus actually uses that strategy as well for some of its proteins, but not for the spike protein. So as far as we know, spike protein only encodes for the spike protein. That genetic code of the virus is only used for that purpose, all right? So that's your little bit of a background on what frame shifting is all about. Now, why are we even talking about frame shifting when it comes to mRNA vaccines? Why would you even be looking at this? And the reason for this is because mRNA vaccines that we used, either Pfizer or Moderna, they did not use what would be naturally found in mRNAs in terms of the chemicals used. They substituted one of those chemicals for something else, for like a synthetic version. And why was this done? By the way, this synthetic version used in the production of these mRNA vaccines, it's called N1 methyl pseudouridine. So basically, it's just chemically modified nucleotide, which normally would be the uridine, and you attach certain chemicals to it, so it's slightly different, and that is used. Why was this used? This altered chemical in the production of mRNAs for the vaccination purposes with the spike protein. The reason why it was used is twofold. Number one, and this is what it was creating problems in the past and why mRNA vaccines could not have been used before until these issues were resolved. Number one is to reduce immune system's ability to recognize it and destroy it. So that's reason number one. And reason number two, it increased the stability of the mRNA construct itself, of that mRNA template. Ironically, both of these issues might lead to its own unintended problems. And I'll talk about that in a moment. But the reason why we employ these strategies is to make sure that the vaccines worked. So basically, because we had a very limited amount of time, we employed all of the possible strategies in order to make sure that the vaccines work. We had one shot only in the midst of pandemic, in the midst of one of the deadliest waves we have encountered in the pandemic, that's when we started mass vaccination. So there was a one-time shot, and there was an attempt to create as robust vaccine as possible. Basically, the purpose was to really make sure we can produce that spike protein 100%. And we succeeded very well. In fact, perhaps too much. And one of the problems we are now encountering is the possibility that the spike protein has been presented to the immune system too much. And this is why we might be seeing such a problem as the formation of the IgG4 antibodies. All right, but that's the reason why these synthetic nucleotides were used. And it was discovered over time that pseudouridines were better than uridines in the production of when you produce the mRNA, in terms of avoiding those two issues, i.e. destruction by the immune system and being unstable, because mRNA can be destroyed very rapidly, and then the additional alterations, the N1 methylation of pseudouridine. Methylation is basically attaching another group of atoms to the already chemical that we had, was even better. So basically, we employed that strategy and we moved on. What might be surprising to you is that despite the fact that we've used this technology with these novel nucleotide bases in the production of these mRNAs that then we used for mass vaccination of the pretty much almost entire world, how these unusual chemically modified nucleotides in the mRNA might affect the ability of the machinery to use that template and produce protein was never investigated. So the question is, can the machinery that it uses mRNA to produce proteins, does it use it correctly or not? That just was never looked at. It was just assumed it will use it correctly and that's it. So that's definitely a bit of a surprising news for sure. And as far as these authors know, they were the first ones ever to look at it. And what they found out is that, number one, indeed, the presence of these modified nucleotides in mRNA does indeed cause frame shifting. And indeed, they're claimed, based on the evidence that they produce, that this also happens post-vaccination with the Pfizer mRNA. That's the only one they investigated. They didn't look at the Moderna one, but it does happen with the Pfizer one. All right, so then now let's get into the details of how did they observe all of this. And they did a really ingenious way of looking at this. In order to determine whether this is working properly or not, they use firefly luciferase. And it sounds pretty crazy. What is luciferase? Luciferase is an enzyme, or basically a protein robot, if you will, because I always think of proteins as miniature robots, if you will. And this tiny miniature robot, luciferase, what it does, it can take a chemical called luciferin and chemically alter it. And after that happens, that chemical luciferin can emit light. Hence, fireflies use this, and hence fireflies, you can see light when they fly around at night or in the dusk. So unfortunately, never happens here in Alberta. But we do have them in Canada because I've seen them. Lovely sight. So why luciferase and luciferin is because it's a play on words. Lucifer in Latin apparently means the bearer of light. Hence the nickname luciferin and luciferase. And it's stuck in science, and now that's what we call. So they use the enzyme. Enzyme means it does some chemical alteration to something else. Protein enzyme called luciferase from fireflies. But they took the genetic code. And they played with it. Genetic code for that to make luciferase. and half of the luciferase mRNA genetic code was normal, good, the way you're supposed to produce it, and then the other half, they put it out of frame. So they basically, the code was +1, the code you would expect, but moved by 1 extra base forward. So that means if the machinery that uses mRNA to produce protein reads it normally, it would only read it normally for about half of the protein. And as a consequence, you're not going to produce properly working luciferase because the other half is out of frame. So then you basically produce a garbage protein, a truncated, fragmented luciferase that cannot do its chemical reaction, and as a consequence, you will not see the light. But if the machinery responsible for the production of the protein, this ribosome, happens to shift itself by 1 spot, then you could potentially produce completely normal luciferase or nearly completely normal luciferase that would still be able to do its chemical reaction, i.e. take that compound luciferin, chemically modify it, and then you can see light. This allowed the authors in this really ingenious way to just simply look for whether if they took the mRNA for that luciferase in that construct and start putting modified chemicals in it, just like it was observed, just like they were used in the Pfizer and the Moderna mRNAs, would they see the shift, which then they would be able to monitor through the production of light. So very, very ingenious. The construct, I think I'm gonna call it a fluke plus F1FS. Basically, that's what the mRNA construct they called. And then the first half that was being coded properly, they called that half as N-fluke, NFLUC, and the miscoded half, the one in Frameshift, that was coded in +1 Frameshift, that was the C-fluke, okay? And that's indeed what they showed, that when you started using these N1 methyl pseudouridines, the same chemicals, synthetic chemicals used in the Pfizer and Moderna mRNA vaccines, indeed, you were able to then start producing light, meaning there was a shift in that machinery, the ribosome, when that machinery, ribosome, used the template with those substituted synthetic nucleotides, and that allowed the production of luciferase, and they were able to now monitor light production. When they just used the normal construct with no messing around, then they were also able to show that just produced luciferase as expected. Someone's shouting behind me. I wonder if that's going to be in the video. Hopefully everyone is okay. All right, so this is really, really cool how they were able to show that. And only that chemical that it was also used in Moderna and Pfizer mRNA vaccines caused that shifting of the ribosome in the use of mRNA to produce this frame shifting. That's the only one. So then, of course, the next big question was, are we going to be seeing this same effect with the vaccines? And they focused on Pfizer vaccine, and basically they wanted to test that with mice. And here also they use fairly ingenious way of determining if that is taking place or not. So they took mice that were vaccinated with Pfizer vaccine, the same Pfizer vaccine that we, the humans, were vaccinated en masse, right? And what they wanted to see is, remember, when you vaccinate, what is the purpose of the vaccine? Is to stimulate your immune system, and for that immune system to have a memory of that stimulation, right? So that's the whole purpose of the vaccine. So therefore, if the vaccine was responsible for frame shifting it would mean it would produce not just the spike protein, but spike protein and potentially bunch of gibberish proteins. And therefore, if the immune system was exposed to these gibberish proteins, then you would code the immune system to recognize those gibberish proteins later on as well. So what the authors did is they basically looked, and how is this immunological memory maintained? It's maintained by B cells, which are the cells responsible for producing antibodies. That's what we're most familiar with, right? Hey, we were vaccinated in order to produce the spike protein. Then when we encounter the pathogen, we expect the antibodies that recognize spike protein to come in and clean up the process, and voila, right? That was the goal of the vaccine. And the other way is T cells. So the same in the same way, when you present fragments of a pathogen or a fragment of an antigen from a vaccine, antigen in this case being the spike protein, you encode that memory in both B cells and T cells. And therefore, and these guys, what they did is they produced a bunch of gibberish proteins as they would expect from the Pfizer mRNA if frame shifting were to occur. And they took the blood of the vaccinated individuals, in this case mice, and they exposed the blood with all its constituents, including T cells, to those gibberish fragment proteins. And if there was immunological memory, then basically they expected that certain T cells that have already been trained to recognize these gibberish fragments, they would be activated and they would start producing specific cytokine called interferon. And they were looking for that interferon. They had a chemical reaction that could spot that interferon. And if the interferon is present, it means, hey, clearly T cells are responding to something they've already been trained to respond to before. And that basically indicates that these mice, when they were vaccinated with the Pfizer vaccine, indeed, they were capable of recognizing gibberish proteins. That means that they must have been exposed to those gibberish proteins. And that's exactly what they saw with mice. All these mice, all of them, they also responded to regular spike protein as expected, but they also responded, some of them responded to those gibberish fragments. What's interesting, they also took mice that were vaccinated with AstraZeneca, and that response was not present at all in those mice. So only mice that were vaccinated with Pfizer mRNA vaccines that used those chemically altered nucleotides in the creation of its mRNA were experiencing this frame shifting effect. So, all right, they saw it in mice, so then they moved on to humans, and they took 21 people who were vaccinated with the Pfizer vaccine, and they took 20 people who were vaccinated with AstraZeneca vaccine, and they did exactly the same experiment. They took the blood, and they wanted to see if we take the blood of these individuals who already had specific immunologic memory, and we provide them fragments of gibberish proteins, as would be expected if you created frame shift using Pfizer mRNA vaccine, do these individuals respond? And the answer was yes. 5 out of, approximately the way I saw from the graph, 5 out of 21 Pfizer vaccinated individuals were responding. Oh, and by the way, how often did they see frame shifting using that luciferase construct? It was about 8% of the time. I forgot to tell you that. So what this all means is that they were able to show that T cells from Pfizer mRNA vaccinated individuals were indeed being activated by those gibberish protein fragments, meaning they had prior encounter to such fragments, meaning these mRNAs from that vaccine in some of these individuals indeed experience frame shifting and production of this gibberish protein. Well, the next thing is my favorite part, is because the authors came up with a solution of how to solve this problem. Very, very valuable. Because remember, this has never been looked for, new technology. When you're using brand new technology for the very first time, yes, there will be new elements that you're going to discover afterwards, that not everything is perfect, because when you use technology for the first time, it's never perfect. Now we have the opportunity to perfect it. And that's why I like that these authors provide a solution. So they suspected, is this frame shifting happening at any specific sites? And they used that luciferase reporter system that I described at the beginning of the video to see if the frame shifting was taking place at any specific sites. And they showed that it does by mutating it back, or changing it back to one of those naturally found chemicals that you would expect to naturally be observed in mRNAs. And they managed to completely solve the problem. That allowed these guys to actually determine the exact sequence that causes frame shifting. And it's very, very simple. It's actually three of those N-methyl pseudouridine chemicals in a row. And by the way, I should have mentioned this, these are labeled as M1 psi, Greek letter word psi, which looks like a trident, and 3 of those in a row, followed by either fourth one or cytidine nucleotide, so one of those that is naturally used. And they were able to show that multiple of these sites are indeed found in Pfizer. So the Pfizer mRNA vaccine for the spike protein. So clearly it means that you could just simply take those few sites, change the sequence a little bit, spike protein can still be produced, but you completely could be removing that risk of frame shifting. And they provided the sequence, so you can see the sequence. What I found is basically that how much of this altered nucleotide, pseudouridine nucleotide, was used in the Pfizer. Basically, I didn't see any normal uridines being involved in the mRNA Pfizer construct. So I thought that was interesting. So solution provided, fantastic, fantastic. But what does this mean moving forward for those who already took the vaccine? Here's where it's a little bit disappointing, because the authors did not address this at all. The only things that they mentioned from when I read the paper, but I did not read the extended data component, just the primary published paper, which, by the way, that paper is published in Nature, one of the most Nature journal, which is one of the most prestigious, if not the most prestigious, scientific journal that there is. Basically, any scientist in the world dreams and fantasizes about publishing any of their work in this journal. Basically, that's the pinnacle of achievement, if you will, in science. So this is not something you can just dismiss lightly. Now, it's only one paper, which doesn't mean much. You need more supporting evidence for sure, but it also doesn't mean that you can just discredit it easily. It's already peer-reviewed paper, and this is one of the reasons why it's stirring so much controversy and so many opinions on the topic. Now, the authors mentioned, like, look, clearly we're seeing off-target immune response. That's what they called it, meaning it's targeting, there's immune response to something else besides the spike protein. Well, makes sense, right? Because these constructs were building something else besides the spike protein. And they were mentioning, well, very likely we will also see similar such off-target response when it comes to B cells and antibodies. And the only thing they mention is that such a off-target immune response could lead to toxicity, without elaborating any further. That's it. The only other thing that they mention is that, well, we do need to improve this. And they, of course, they provided the solution. And the reason why they mention is that because we need to make sure that the future iteration of such the mRNA technology as was used for vaccination this time, we have to avoid these problems, because this frame shifting could be potentially serious problem if more antigen is needed to be presented. And which made me think, like, really, even more antigen? The way I'm seeing and interpreting this information is that one of the problems we ran into is presentation of too much antigen, because we created mRNA vaccines that were so amazingly efficient that basically they ended up being more efficient than we ever thought they would be. That's why we went for such an overkill. And that's why basically every single uridine was substituted with this synthetic uridine to make sure that these mRNAs are very stable and are not easily recognized and destroyed by the immune system. So that, I thought it was really interesting. And the example they used of how such mRNA technology could be used in the future was for production of hormones, where you might require more antigen and more frequent antigen presentation. I thought that was interesting. But that was it. They didn't really elaborate any further on as to what the problems might be. Now, what are the problems? Well, as I mentioned, I've had the privilege of working with a number of different scientists where basically we are looking at the mRNA technology as it was used for the vaccines, and we're analyzing what could be the problems. And the reason why we're doing is to basically, indeed, to make sure that if problems are uncovered, we can think of solutions to make sure that the technology can be improved in future reiterations, right? That's the whole goal. We're not talking about of not using the technology. Obviously, all of us are proponents of smart use of vaccine technologies, but we need to also know how to improve this technology and not use it in a dumb way, basically. So we've been working on a number of papers for that purpose, and one of the latest ones that we worked on was the influence of these chemically altered uridines on health. And the potential problems, I won't be discussing the details here, is we did mention is potentially the frame shifting. Now, if frame shifting does happen, what could it be leading to? And in that article, we were addressing the possibility of maybe it could aid cancer development, because there is some emerging, so this is not written in stone, emerging information that perhaps cancer could be utilizing frame shifting in order to evade recognition from the immune system, right? But this is hypothetical, so that's one issue. The other one, far more obvious, is that if you produce gibberish proteins and you present it to the immune system, and the immune system is now recognizing it and producing a response to that gibberish, well, what else could that response be recognizing besides the gibberish protein? Well, what if it starts to recognize something else in our own body? What if that component of that gibberish we created happens to mimic something that is found in our body? And there you go, you could be inducing autoimmune conditions. So these are the more obvious ones. But the other one that also I thought I would mention is the concept of aging. So why aging is because misfolded proteins, or proteins that are produced out of frame, what I've just been calling throughout this entire video gibberish proteins, that can happen naturally, and it does happen, and it can be observed that such proteins can be accumulated in the process of aging. Such abnormal proteins, they can aggregate, and they can lead to neurological conditions such as, say, for example, Alzheimer's or Parkinson's. Okay, so there could be a problem when it comes to aging. Now, that all sounds scary, but the same group of authors, and believe me, these are very, very smart individuals. I basically feel like an apprentice, scientific apprentice, when I have the privilege of interacting with these individuals, because these people have many years, decades of experience of looking at scientific problems. So they have a wonderful capacity of seeing things in a bigger picture than sometimes I can. And that's the big pleasure. And they mentioned that, look, clearly we're producing proteins all the time. We can mess up production of proteins as well, and we have a built-in mechanisms, very robust built-in mechanisms that normally supposed to take care of this. So we have systems in place inside our cells that clean up this process. They clean up the misfolded process. But as I mentioned, sometimes that can actually be overcome. And the overcome as in the system might fail if there's too much of a problem presented. And that can, so in healthy individuals, these group of scientists are saying maybe healthy individuals, they don't have to really worry about anything because the mechanisms in place will protect them well enough from, even if they do produce any gibberish proteins as a consequence of vaccination. But those who say might struggling with their health, such as cancer individuals or aging individuals, their cleanup systems might not be robust enough in order to actually succeed. And perhaps that might be the individuals that are at higher risk. All right, well, maybe then we need to wrap this up with, why did this happen? And is there anyone to blame for this? Because this is actually much more challenging question to answer than you might think. It's very easy to think, oh, to point finger, to blame any specific institution, but it's not always necessarily so simple. And here's the concept. Well, why did it happen is because we didn't have time to analyze this information. The reason why we didn't have time to analyze this information is because we were so keen in being able to bring the product to the market so that we have a solution under emerging health threat, right? So this is the reasons why it happened on such a rapid scale. And when you have to produce vaccine in crazy record time, you're not going to be able to investigate everything. And that's exactly what happened. But is there anyone to blame? And you could, well, look, the way I see it is that the industry did the minimum possible what they were required of in order to get this available to the market as fast as possible. So then would the regulators be at fault? And if there is anyone that I would be pointing fingers, it indeed would be the regulators, not because I would be placing any fault at them, but that's probably where we have to start thinking of how to improve the processes in the future. And the reason why is because it's the regulatory bodies that are obviously, they have the power invested in them in order to protect the well-being of the public, right? But here, under these circumstances, the regulators were also under insane pressure to make sure that the vaccine comes to the market as fast as possible. Basically, everyone was demanding it because we wanted such a rapid solution. So are the regulators to blame as well? And I also don't necessarily think so. Like, imagine, put yourself in their position at the time where everyone was panicking. So if there was one entity that I think we need to really reconsider, is the role of the media, because it's the media that has potentially over-exaggerated the threat of the potential disease while underplaying any potential risks of the technology, which was brand new and never used before in humans, right? So that was not specified enough, while the threat was overblown to potentially record proportions, not to diminish the horrific negative consequence of this pandemic, because millions of people died. But nevertheless, that's what happened, right? Like, there was, as a consequence, the public was extremely hungry for a rapid solution, and that rapid solution seemed to be vaccine. So there we go, we produced vaccine, but because of how fast we produced it, we were not able to test everything about the safety of these vaccines. And now we are slowly, years later, discovering some potential problems. Potential problems because, like I said, We don't know to what degree this is still an issue, but we now have three major potential issues that need further investigations. And number one is the IgG antibodies, IgG4 antibody formations in the mRNA vaccinated. Number two is the DNA contamination in these vaccines, and what will be the biological consequences of that, which, by the way, we still don't have any peer-reviewed content to corroborate that information. And then now third one, the potential of frame shifting taking place because of how the vaccines were constructed, right? So there's a lot to now unpack, but the part that I really like about this particular information is that at least we have an easy solution moving forward. Same thing with IgG4, perhaps we have an easy solution moving forward, which is basically reduce the antigen presentation, because we might have accidentally produced vaccines that were too good in producing the antigen, and it presented it too much of it to the immune system. So to some degree, seems like we'll be able to overcome some of these issues at least. So that's the good news in all of this. Of course, for those who are mRNA vaccinated, well, that doesn't mean you need to panic. As I mentioned, based on the conversation with the scientific small scientific community, it's likely those who have already other comorbidities and are not in the ideal health condition that maybe might experience issues, which is only hypothetical, and the vast majority of people should be completely fine. And based on the results of this particular paper, only a minority of the vaccinated individuals with the Pfizer vaccine were producing immunological response to off-target gibberish content. All right, that's a long video. I warned you there's gonna be a longer one because of how big and monumental this was really, and how interesting the science is. I was gonna break it down into greater detail. As always, thank you for all of your support. Thanks for all the questions coming in. Thanks for sharing the videos. This is how we grow, and the channel is always ever so slowly, tiny bit by tiny bit, growing month after month. So that's wonderful. Thank you. And yeah, please keep it coming. And I look forward to seeing you in the next installment. Bye, everyone. Ciao for now.